Today, we are diving into one of the most important papers in modern biology, a piece of work that fundamentally changed how scientists and doctors think about cancer. We're breaking down The Hallmarks of Cancer by Douglas Hanahan and Robert Weinberg. All right, let's get into it.
Think about that for a second. For decades, this was the central problem. Cancer isn't just one thing. It's this huge umbrella term for hundreds of diseases. They can show up in any organ, they behave differently. It just felt so impossibly complex. Where do you even start looking for a common thread?
And hey, don't just take my word for it. The authors of the paper said it themselves. By the year 2000, when they published this, cancer research was just a mountain of data. An incredible amount of information for sure, but it was so fragmented it was getting really hard to see the big picture.
And this is where the genius of their idea comes in. They just flipped the question. Instead of getting lost in all the differences, they asked, what if we look for what's the same? What if there's a basic set of rules, a set of capabilities that every single cancer cell has to acquire to become dangerous? And they found them. Hanahan and Weinberg argued that all that mind-boggling complexity could be boiled down to just 6 essential changes. 6 rules that a normal, healthy cell has to break on its journey to becoming a full-blown cancer cell.
So here they are, the 6 hallmarks. You can think of them like 6 major hurdles that a budding tumor has to clear. And each one of these represents a breach of a really powerful anti-cancer defense that's already built into our normal cells.
So let's actually walk through these 6 rules, and we'll start right at the top with number 1. Okay, so normal cells are team players. They're polite, they wait for signals from the outside world to tell them, okay, it's time to grow and divide now. Cancer cells? Well, they decide they're done waiting for permission. They figure out how to make their own grow signals. This is just the perfect way to think about it. The cell is essentially hot wiring the system so it can press its own gas pedal. It's not waiting for the driver, which is the body, to give the signal, it just floors it, creating this constant, relentless drive to make more and more copies of itself. So how does that actually happen? Well, one of the most common ways is through a mutation in a gene called RAS. Now, the RAS protein is a crucial switch in that go signal pathway. When it gets mutated, it's like the gas pedal is physically stuck to the floor. And that one specific mutation, it's found in about 1/4 of all human tumors.
Right, so having a stuck gas pedal is one thing, but it won't do you much good if someone else is standing on the brakes. Our tissues are full of stop signals to keep everything in check. So to get anywhere, cancer cells have to figure out how to completely tune out those commands.
And there's the other side of the coin. The cell literally cuts its own brake lines. There's a master brake in our cells called the retinoblastoma protein, or PRB for short. And it turns out that in the vast majority of human cancers, that PRB braking system is broken in one way or another. The brakes are gone. There is nothing to stop that constant go signal. Now, our bodies have this incredibly important safety feature called apoptosis. It's basically programmed cell death. It's designed to get rid of cells that are damaged or just not needed anymore. So if a cell starts acting weird, like dividing when it's not supposed to, it should trigger its own self-destruct sequence. Cancer cells absolutely have to find a way to disable this.
Yeah, for a cancer cell, this is all about survival. One of the main things it has to shut down is a famous tumor suppressor called p53. You might have heard of it. It's often called the guardian of the genome because it can trigger that self-destruct sequence when it senses DNA damage. So it should be no surprise that more than half of all human cancers have a broken, useless p53 gene.
Most of our normal cells have a built-in lifespan. They have a counter. They can only divide a limited number of times, maybe 60 or 70 times, and then they just stop. This limit is physically tied to the tips of our chromosomes, which are called telomeres, and they get a little bit shorter every single time a cell divides.
But to build a tumor big enough to be a real threat, cancer cells need to divide thousands, even millions of times. So they have to find a way to become immortal. And they do it by switching on an enzyme called telomerase. Telomerase rebuilds those chromosome tips, effectively resetting the countdown clock and letting the cell divide forever. And you find it active in something like 85 to 90% of all cancer cells.
So, a tiny little clump of cancer cells can get by just fine on its own, but once a tumor grows bigger than, say, the head of a pin, it hits a wall. It needs its own dedicated blood supply to bring in oxygen and nutrients. It has to convince the body to build it some new plumbing.
And that's exactly what it does. This process is called angiogenesis. Tumors start sending out chemical signals. A key one is called VEGF, that are basically construction orders for nearby blood vessels. They trick the body into building new branches that lead right to the tumor, feeding it and helping it grow. And that brings us to the last and, frankly, the most deadly capability. See, the first tumor that grows in one spot is rarely what kills a person. The real danger, the thing that makes cancer so feared, is when cells break away, get into the bloodstream, and set up new tumors in other parts of the body.
To pull this off, the cells have to learn to cut ties with their neighbors, break through physical barriers in the tissue, and survive a journey through the bloodstream. And this is the most critical hallmark because of one sobering fact. This process, metastasis, is directly responsible for about 90% of all human cancer deaths. 90%.
So let's just recap that really quickly. Press your own gas pedal, cut your own brakes, ignore the self-destruct commands, become immortal, build your own supply lines, and finally escape and colonize new territories. Every single successful cancer has to figure out how to do all 6 of these things, which leads to a pretty obvious question, right? How on earth does one cell get all of that done?
Yeah, that's the multi-billion-dollar question. How does a single cell manage to acquire all 6 of these incredibly rare and complicated abilities? The odds against it should be astronomical.
Well, the answer lies in something the authors call an enabling characteristic. The cancer cells break their own DNA repair systems. See, normal cells are amazing at fixing typos in their genetic code, but cancer cells lose that ability. Their genomes become unstable, which means mutations start piling up way, way faster. It essentially turns evolution into a high-speed process inside the body, generating enough random changes to eventually stumble upon the mutations that unlock these 6 hallmarks.
The paper ends with this really powerful vision for the future. They argue that if we understand these hallmarks, we can stop thinking of a cell as just a random bag of chemicals and start seeing it for what it is, a complex, integrated circuit. And cancer isn't just chaos, it's a logical, if deadly, reprogramming of that circuit. And if you understand the rules of the circuit, you can start to figure out how to fix it.
And this brings us to the ultimate takeaway, the reason this paper changed everything. The Hallmarks framework shifted the entire goal of cancer therapy. What if, instead of attacking a lung cancer or a breast cancer based on where it is, we could develop drugs that attack angiogenesis, or drugs that force cancer cells to self-destruct again? We could target the fundamental rules of the disease, no matter where it shows up in the body. And that one simple idea is still shaping the future of cancer research today.